Hi, this is Dr. Ben Morrill. Welcome to episode 130 of Reptile Genetics Weekly. Glad to have you here. We've got some fun subjects to talk about. We'll kind of wander a little bit with a few different things, but we'll start out as we mentioned last week. We're going to talk about orange ghost versus hypo, and we'll also kind of touch on all the other names there were associated with ghost/hypo and ball pythons early on. So talk a little bit about that and why maybe that was the case and what's going on genetically, and also kind of talk about some other projects, dinkers, different things, and end up by giving you another snippet of what we plan to talk about next week. So, but before we move on, I will do sample update. We should have fast tests going out like normal every day this week. Same with sex determination. The panel testing, I told you all that I hoped that those results would go out over the weekend unless something bad happened. Well, something bad happened. It's been a really crappy weekend for me. I've been doing a lot of stuff all through weird hours to get this crap working, but it's still broken. So I am right now, actually, while we were getting ready to record, is working on that. So we hopefully that will go out probably on Wednesday. Wednesday, Thursday, Friday, we'll have results going out. So sorry to all of you that are affected by that. But the good thing is, once I do have this locked in, it will make panels be faster. We'll make it so turnaround times will be half to like a third of what they were in the past. So it sucks right now. It really sucks right now. But it does look like it is going to work and be really, really good for turnaround times, make that really good. So if you're doing a full panel or like a spider panel or something like that, you don't have to wait like 4 or 5 weeks. It'll just be like a couple of weeks. So, and maybe even get that down to 1 week during the busy part of the year. So that's what we're working toward. And sorry and thank you once again for your patience. I know many of you have messaged me and Sean asking about that, and I promise it is coming. I'm working on it all the time, and it's getting close. So hopefully Wednesday, Thursday, Friday this week. But yeah, that's what I've got for updates. Kayla, how are you doing?
Hey Ben, I'm doing good. I appreciate you working away at this. Is it kind of a similar situation to what Sean was describing, or at least the metaphor he uses is like take a margarita and add 73 more ingredients to it, but also make it taste the same. Is it kind of like that a little bit?
Potentially, yes.
Potentially. Okay, fair enough. I know
my,
that's kind of how he
my
described
brain's
like
fried.
what's that?
I said my brain's pretty fried. To
That's
me,
fair.
there's 1,000 things I'm thinking about. But yeah, it's
Yeah.
essentially like trying to make something using all U.S. metric kind of or U.S. types of tools, and now you want to try to make the same thing, but you're using different tools that aren't the same, but they're supposed to be similar, and trying to
Oh,
make
very
all the
good.
tools work right. So the
Mm-hmm.
the cocktail that Sean was talking about, that
Yeah.
part should still work the same. This
Okay.
is all the other parts of the process that have to get working a different way, and they're not quite there yet, but it's close.
Fair enough. All right. So just so y'all know, we are going to Tinley this weekend. It's going to be my first time at Tinley. I'm so excited. My best friend Krista's coming along as well. So you'll find the three of us at the RGI table. We'll be outside in the hallway, and I'm so excited. Excited to see everybody. So, be sure to stop by, give us your research sheds if you've been meaning to mail them in and have been sitting on them for weeks or months or years, meaning to send them in. This is your excuse. This is your sign. Bring them to us at Tinley. Um, yeah. And today we actually have a guest, our friend of the show, Dr. Travis Wyman, our friendly local mad scientist, here to join us and talk about the hypo ghost and orange ghost situation and how on earth you have two different well-known morphs, but it's just one mutation that we can test for.
Yes, and we've got him talked into. We're all planning to do this again next week. So Dr.
Yes.
Travis Wyman will be with us next week too. So that'll be good.
Yes, yes.
Yeah. All right. And for people who aren't familiar with him, which he's here all the time, you should be. But in case it's your first one, or you just happen to miss all the ones that Travis was in before, Travis is a geneticist and fellow reptile nerd. So we love to kind of wind him and Ben up and just let him go, and it's great. Good things happen. So here we go. Hypo Ghost and Orange Ghost. So for people who are, I don't know, colubrid nerds like me and have no idea what's going on there, what's the deal with Hypo Ghost and Orange Ghost?
I would say before genetic testing, the two most common terms that people would use to talk about hypos, really the more correct term. Calling it a ghost is kind of confusing when you compare that to other species and how we use the term ghost. So
usually it's a combo of some kind, like a morph combo, like axanthic and albino, maybe.
Yeah.
Actually, I think that's usually a snow. Anyway, usually known as a morph combo.
It was a axanthic hypo is what was termed ghost throughout every other aspect of the hobby, but then ball python people had to be weird.
Many such cases, and that's okay. We love them for that.
So it's a little complicated, but yeah. So hypo, people did eventually get to where they're using hypo and ghost interchangeably, but they would still call some of the animals orange ghosts, and kind of thought that was a separate thing. But once we started doing the genetic testing, both us and Dr. Hannah Sadell ended up publishing a paper on it. But it was very clear that all of these hypos or ghosts seem to be just one mutation. And we've seen a few rare instances where people have hypo ball pythons that don't test properly with this current test we have. The vast majority of animals that are ball pythons and are hypomelanistic, they are this same -5 mutation that Dr. Sadell and her students found.
Right. And so, and as far as those like anomalous, definitely a hypo animal, but doesn't test for it, those are really like probably not related to today's conversation because you've tested probably hundreds or thousands of hypos at this point.
Yeah,
right?
by far and away, these are. But if you yourself, if you ever have a hypo and test negative and you want to dig some more, a couple of people we've talked to that had this, they're like, yeah, they didn't have very many, and they're like, I'll just not breed them, and I'll use the one that you can test for instead of trying to make more of the one that doesn't test right and trying to figure out a test for it. They would rather just use the hypo is common enough in ball pythons. They didn't want to complicate things. So there wasn't really a reason for us to design it. But if you happen to have several hypos in your collection and they don't test properly with the current test and you want to chase that some, reach out to us. And that's something that we like doing. We might not be able to have a lot of time to do it because we're doing 1,000 other things, but that's something we do like playing with when we can.
For sure. We love teasing out those mysteries.
But yeah, by far and away, vast majority, they're all this -1 mutation.
Okay, cool. So we got for example, this is our karyotype of chromosomes here. Ball pythons have 18, I think, but we're just going to go with 10.
So our bouncing chromosome, we didn't prep Travis specifically for this, although he can make it easily, I'm sure. But the bouncing chromosome, that's just like we talked about last week, that's our chromosome we're saying has this hypomelanistic mutation.
Yep.
The MLPH gene is what's mutated. And so this is just showing where that mutation is. So out of all of those base pairs, what's a common size for a chromosome, Travis? Dozens to hundreds of millions?
Yeah. I mean, again, it's going to depend on the chromosome, and some are really big and some are really small, but yeah.
Okay.
Yeah, we're talking millions, and this is just one gene of that chromosome, so it's just a very small portion of it.
Yes. Oh, I made this one wiggly too. Well, it's kind of cute actually. So one gene in one chromosome and one little mutation on that gene is what causes what we call the hypo or ghost mutation, including orange ghost. on the MLPH gene. What is that actually like? What is that abbreviation for?
Melanophyllin.
Melanophyllin. Okay.
There you go.
Yeah.
The green there, that's the protein, the melanophyllin protein, the code. We get a -5. It makes that green part of the C figure there, so it doesn't function properly. So that means that the melanosome, which gives it the color, doesn't have the opportunity to hook up to that actin filament at the bottom, and that's what is kind of like its roadway. And so, what was the term you used, Travis? This makes it so it doesn't travel properly.
Melanin deposition dysfunction.
There you go.
The melanin is produced, but it is not deposited properly in the body.
Mm-hmm.
Yeah, so it's kind of like you have cargo you want to transport, and you've got a road to drive it on, but you don't have a way. Melanophyllin is kind of like the car that you would put the cargo in and then drive on the road. You don't have a car. You just got the cargo and a road to travel on, but you don't have a way to actually transport it.
I look at it more like the tow hitch.
The tow hitch. Yeah.
If you've got your truck and it's got a tow hitch, and then you've got a trailer, and you got to hitch them together. If that tow hitch, like if the ball doesn't fit right, then you can't hook that trailer on. And if the trailer's full of melanin, now you can't haul that trailer where it needs to go because your tow hitch isn't right.
Yeah.
That's great. I like that.
I like it too. And I think that's kind of what blew people's minds when we did an episode on that, which it should pop up like at some point if I don't already have it thrown up. The thumbnail for that episode, or the title, was like, the hypo or ghost mutation doesn't actually reduce melanin production at all, despite, hypomelanin. You would, like, since that's what it's short for, hypomelanistic, you would think that it reduces melanin production, but it actually produces the same or the correct amount of melanin. It just isn't distributed right, as is shown in this little table here. This is what's hypothesized and pretty well understood to be, you've got your melanin. It's just not being distributed, and that's how we get that ghostly hypo look that we love so much, which is just so freaking cool to me. I love it. We're going to kind of step to the side a little bit, but we're going to wrap back to our orange ghost conversation. But let's talk about how you can have your own collection can be genetic within itself, and what we mean by that. Yeah.
So if we look at these animals here, thanks Living Art for, making them available to us.
They're so pretty.
We see, our two striped animals, the male and the female. And when bred out, the animal on the right comes out, which is, while somewhat striped, is definitely not as prominently striped as the parent animals were. So what you can take from this is, just because you see something does not automatically make it a true single gene morph. But that animal on the right still has a lot more striping to it than what you would consider a normal wild type animal to have. So there are a number of genes that can contribute to striping. And that animal, the parent animals have probably a bunch of them, some of which may be recessive or incomplete dominant. And that rightmost animal doesn't have necessarily all of those genes, because of, the way your genes segregate out in the gametes. And you can see this with all kinds of things. I know that I have mentioned in many of my past episodes, I really like stripe animals. So a lot of my animals in my collection will produce very stripy looking babies, because, if something is more striped than its sibling, but they're both, if I produce a clutch and I get two of the same combo and one is more striped and one is less striped, I'm going to hold back the more striped one because I like stripy things.
Yeah.
Now, there are other people who don't like that. There are people who like banded animals, so they're more likely to hold back the banded animals from their clutches. There are people who really like high blushing animals. So they'll hold back animals that are really, really high blushed. We saw that decades ago when we had pastel and we had super pastel, and all of a sudden we had super duper pastel. And we had pastel and we had super pastel and we had whitewash pastel. Well, what was happening was people were holding back their highly blushed animals and continued breeding them together until all of a sudden they got these super crazy, ultra blushed out pastel animals.
Yeah.
And they thought that they had something new. Well, I mean, yes, they did, but what they had new was not new so much. It was that they just kept enriching those blushing genes in their own personal collection. And that's what brought it to the fore. Then later they sent all those animals out, and all of a sudden they no longer were making super duper pastels or whitewash pastels because those high blush genes were getting washed out by going into other people's collections.
So the things that we select for either knowingly or unintentionally, we just think, oh, that one's prettier. I think this one's prettier than the other. That's what we end up enriching more and more of in our collections.
Yes, and that makes your collection genetic in as much as you are enriching for genes.
Yes.
It's just not necessarily specific morphs that you're enriching for. It's just genes that one way or another you have a preference for. And we have done this throughout history. I mean, there's a reason that, you drive past a field of cows and they're modeled black and white. And then you look at the dog sitting next to you and it's modeled black and white. And then you look in your freezer when you go to feed your snakes and you have rats that are black and white. That those are all types of piebaldism. And people like piebaldism. It's more attractive to them for some reason. So piebaldism is something that always pops up in domesticated animal populations because people like things like that. Floppy ears in dogs. Look at a wolf. Look at the dog sitting next to you. Odds are the dog sitting next to you is going to have floppy ears. Maybe not your specific dog, but labs all have floppy ears. Beagles, bassets, they all have floppy ears. Again, you've enhanced the pool for that genetics because you liked that. I think dogs also have like extra bones in their tail to make it longer and make it more flexible, which, we like that because they wag their tail.
Because they got flappy tails,
Yeah, there are all kinds of things that we do like that.
Right.
And, they didn't do it on purpose. Yes, you can intentionally breed for piebaldism. But how do you intentionally breed for a dog with a longer tail? You don't. You just notice that some dogs, puppies, have flappier tails. And those are the ones that you think are cuter. So you keep those puppies back. And then all of a sudden, thousands of years down the road, hey, look at this. Turns out that dogs have more vertebrae in their tail than wolves do. It's just a weird quirk that happens.
And so we do the same thing with our animals.
Yes. And again, if you go back to the way back, the early histories of ball pythons, a couple decades back, when forums were a thing, before social media was the thing, there were a lot more than just orange ghost. We had butterscotch ghost, and peach ghost, and yellow ghost, and green ghost. And basically every one of these was a different animal that probably was picked out of a wild-caught bag lot and looked a little bit different. And then when they started breeding it within their own collection, they were inadvertently selecting for the things that they liked better. So peach ghosts, orange ghosts got the name through Nerd because they hatched out and they had this kind of orange hue to them, only as hatchlings, mind you. As they turn into adults, they all look the same.
Really?
Peach ghost, those came out of Ralph Davis. He liked them because they had a peachy color to them. It wasn't orange, it was peachy. Ralph was really strongly adamant about that.
I don't know. I'm looking up pictures now while we're doing this.
Yellow ghost, butterscotch ghost, all the same thing. The babies looked just a little bit different. And so people started selecting for those just a little bit differences and then it diverged enough that the babies did look different from one another. But ultimately, it was the same mutation, and the reason they looked different was not because of the mutation. It was because of all the other genes that were hitchhiking along that you were picking for.
Yeah, and so when you compare to last week when we were talking about ultramel, for example, where we have two different mutations that cause one morph. Well, in this case, we had what we thought were maybe multiple morphs, at least the two, the hypo and orange hypo. But, like Travis was bringing up, if you go back even further, you've got butterscotch and peach and yellow, all of those. So for a little bit, people thought maybe those were all different morphs, but really those are all just one mutation. So it's potentially 4 or 5 or whatever morphs we thought back then, but now we know it's all just one mutation. And likely those small differences we saw when they're hatching out are differences in our collections. So having that, genetics, our collection being genetic, like Travis was talking about. And talk about the other term I liked that you used was hitchhiker genes.
Hitchhikers, yeah.
Talk a little bit about that, Travis.
Yeah.
And it's, again, like I said, it's not the genes that cause the mutation. It's the other genes that are hitching along with you. So this is an animal that I produced here. It's a crazy looking animal. And if you look at that, you immediately like, there's something special here. Okay.
Yeah.
I will tell you, one of the parents of this animal is a het monsoon, genetically tested, proven het monsoon. Now, if you're in the monsoon project, if you're in ball pythons, you've probably heard a lot of people referring to the hets for monsoon being visual. And it's things like this that look like this. But if you talk to more people, there are a lot of people who buy animals that look like this, this crazy whacked out pattern, the funny head stamp, just the busyness of it.
Yeah.
And they buy it based on appearance, and then they'll test them, and they're not het monsoon. And like I said, the parent for this one is proven tested het monsoon. The sibling to that parent, a friend of mine owns, looks the same. The parent looks like this too. The parent of this one looks like this too.
Yeah.
The sibling that my friend has looks like this. He's produced a whole bunch of animals. A whole bunch of them look like this. He's had them tested. None of them are het monsoon. So what may be going on here, and again, this depends on who you talk to and how big of an argument we want to get in, is that monsoon is not a visual het. But Dave, when he was, breeding them initially, he really liked busy looking animals, or he really liked busy looking monsoons. And the more busy looking monsoons were the ones that he kept breeding up. And so these genes that make it look all crazy busy like that hitchhiked along with the monsoon gene. And now that they're getting out of Ben's collection and into the greater, hobby at large, you're starting to see these busy pattern genes come leaking out, and they're moving along on their own. And we see this all the time. When Ralph let Blackhead out, everybody's reporting like, well, my blackhead looks different than your blackhead. My blackhead's got this. Some of the babies from my blackhead are, like the reddy orange. Some of mine have got these weird ring looking side patterns that they're not normal. Well, that's because Ralph was picking these things that he liked. And what eventually happened was when he let him out, a lot of his animals had hitchhiker genes. And those genes we have later come to call the red gene and the ringer gene. They were just piggybacking along with the blackhead and Ralph's animals because of what Ralph was picking. And once it got out, you started seeing all this craziness. Blackhead being an incomplete dominant gene, it's a lot easier to explain and understand, well, there's other things in there, and people got that real quick.
Yeah.
With monsoon being more recessive, people immediately jump on the argument of, well, that makes it a visual het. Well, no, not necessarily. What it means is there's things that have been moving along with these animals for a long time.
Yeah.
And now you're starting to see them in their own light. And maybe no longer piggybacking like my friend who got a crazy busy animal but is proving to have not been het monsoon at all. And he picked it up based on the appearance alone with the argument of the hets are visual. So if you get an animal like that, you know it's going to be a het.
Yeah. It kind of reminds me of so like in German Shepherd dogs, for example, there's a lot of talk for any breeding dogs, you're talking about conformation of an animal, like conformation. German Shepherds a lot of times have that like sloping hips that give them hip dysplasia later on. And like, there's a lot of genes that control that sort of conformation. I assume, I could be wrong. Or in horses, you're going for, like the withers, the shoulders, and the hips. You want those to be even. You don't want one to be higher than the other, or, the butt to be too big, or the profile of the face to be too cupped or too bowed out. Hello, Potato. The cat is here. It's not that your animal is homozygous or het for a cupped face profile. It's that if you have an animal that has good conformation, a mutation that you think looks good, you're going to select for that. You're going to breed that animal, hope for, and then select from its offspring that look closer to it. It's not het for good conformation. It just has hitchhiker mutations that you are selecting for over time. And so I think in the world of ball pythons and snakes in general, we're very trained for, is it het? Is it homozygous? Is it not? And I think we need to get out of that, take the blinders off of that a little bit, and allow for the complexity that is what creates your animal, I guess.
Yes. And I, as I so often do, I will kind of blame I don't know if blame's going to go over well, but I'll own this. I kind of blame the founding people in ball pythons and their genetics with they had a very, very limited understanding of genetics, but because they were the founders, everybody took their word as gospel. We need to stop just doing that. We need to own the fact that the people back then didn't know what they were talking. I mean, this is science, people. This is what scientists do all the time. Originally, scientists believed that if you took corn and rags and stuck it in a corner, it would turn into rats. I bring that up now, and everybody's like, what kind of stupidity is that? Like, okay, scientists were all like, if you take the corn and you put it in a locked metal box and you put rags in there with it, it doesn't turn into rats. Do you know why you have rats? Because the rats eat the corn and you're leaving it out where they find it.
Like, yeah. They don't appear from nowhere. But that was a belief that they could.
Yeah. We can evolve. We can say, look, they came up with explanations back then that made sense to them at the time. But, times have changed. That was the late 70s, early 80s, late 80s. Come on, people. Think about this stupid device that you have stuck to your hand right now. Think about what those looked like back at that time. They were stuck to the wall. They had a cord on them. You had to pay all kinds of money to call anybody anywhere else. You didn't have video capabilities. Like, think about how far this has evolved. That's science that has caused this to evolve. And science has caused everything to evolve. We have a much better understanding of the genetics and everything that we do with our snakes. And that's what Ben's over here doing. There we go. That's what Ben's over here doing. Yeah. He's harnessing this technology and our understanding to give us these answers. We have that information now. Put away the garbage. It's okay to say it was wrong.
And we're building on the foundation that they helped lay and communicating the new evidence and new science that is built on top of a lot of that. And
Yes.
sometimes that means saying, oh, they actually weren't quite right here. Actually, it's this. And that's the same thing that happened with, it's not even to say the butterscotch ghost, peach ghost, yellow ghost, green ghost, etc., and all the other various hypos that appeared at the time were incorrect. It's just that, we thought they were different things and realized later on that, oh, actually, the mutation we're selecting for is one mutation plus a whole bunch of pretty hitchhikers that we select for intentionally or unintentionally. Hello, potato. And that's just how we move along and figure out what we've been selecting for in our genetics, in our collections, and so on.
Definitely standing on the shoulders of not only the early pioneers breeding in reptiles, but also the scientists that did this work in fruit flies and snapdragons and all kinds of other things.
Zebrafish. Oh yeah, for sure.
Mice,
yes.
Yeah, so it's a lot of stuff, but I know, and I've said it many times too, it's like, but people have been calling it that for this long, it's going to be hard to change. And that's true. It is hard to change, but we can.
But we can. It's okay.
We do have good information that makes better sense. And I think one of the most important reasons why, like, especially if you're a breeder and you want to be able to make money breeding reptiles, there's really, when you talk about supply and demand, you have to be able to make enough money, there has to either be less supply, so what you make is more money, or there has to be more demand. And so if we use terms that are more likely to make sense to new people, if they're coming from dog breeding or coming from horse breeding or something else, if we use terminology that makes sense to them, then it's more likely that they're going to move into this and feel comfortable than if we're using terms that only, reptile or ball python people understand. They're going to come and be like, oh, this doesn't make any sense. This is really confusing, and then be less likely to want to be a part of it. So I think that's another really good reason to update the phrasing that we use to kind of match what we understand as breeders, but then also from science, from genetics.
Yeah. Well, and building off of that, you also have when we have things where we're having to argue our side that we're not a bunch of just crazy reptile people. Reptile people are not crazy. We don't feed live animals to our snakes because we're all sick and twisted mentality. Like, again, if you have to go into a formal setting and discuss this, as has happened numerous times with all the reptile bans and all the different state and locality bans and stuff, if you come in there and you talk in a professional and intelligent manner using the right terminology, it's a lot harder to dismiss you as being a crackpot.
Mm-hmm.
If you go in and you're just using gobbledygook words and made up terms, in some ways you're kind of feeding into that stereotype of these people don't know what they're talking about. Mm-hmm. We can't trust them.
Yeah. It legitimizes what we do.
Yes.
And that's important, especially when reptile bans for very silly reasons start coming through. Hurts the hobby as a whole. So I think it's kind of like we said last episode. This is a much broader subject in general, but we're not being pedantic. We're just trying to make it accurate. Yeah. But
yep.
Cool.
Give you the information that you need. You can spread the word.
Exactly. So next week, speaking of, kind of getting our lingo up, I guess, and also explaining some of building on the foundations of the original subjects we learned about. Next week, we're planning on talking about the 33% possible supers for our dominant traits like leopard or tessera in corn snakes. So you guys
yeah, you'll have to watch. It's just a teaser. We're not telling you much right now, but
Yep.
we'll have
Travis will be there.
Yes, we'll have a good conversation about that, and we may even, I don't know if we'll be able to have someone from Morph Market on the show. We may, but if not,
nice.
we'll have some input from them as well, and also look forward to hearing how you all what you think of this idea, and if you think it's a good idea. So definitely come back next week for that conversation.
Oh, there's a common yellowthroat warbler outside. I'm so sorry. I've become a bird nerd, but like that's my lifer right there. That's my life for common yellowthroat warbler. What's up?
That seems to be a thing with herpers is, you start keeping reptiles and then you start going out looking for reptiles. And then when you're out looking for reptiles, you suddenly realize there are other animals, and it's like a whole bunch of them go the bird route.
Yeah, birds or like entomology, seeing bugs or
yes.
It could be amphibians. Yeah.
Oh yeah, for sure. Yeah, no, and yeah. The world is so cool.
Yeah.
Yes.
All
Cool.
right. Well, thanks everybody. Let us know if you're excited for next week's episode. Let us know what you thought of this one. Maybe clarified some things or just created more questions for you. We're okay with that too. And of course, like, comment, subscribe. All of that helps us in the algorithm. Tells everybody that we're cool and definitely worth checking out. We think we are. And yeah, y'all got anything before we call it a day?
That's it for me.
Awesome. Travis?
Nope, I'm good on my end.
All right. Well, thanks everybody. We'll see you next week.